In this module, we'll talk about the structure of the human genome, as well as different types of DNA variants and mutations. The human genome comprises 23 pairs of chromosomes. All of these chromosomes are extremely similar from person to person, 99.9% identical. The relatively small number of differences is what makes each person unique. One of the pairs of chromosomes are the sex chromosomes, XX for women, XY for men. The other 22 pairs of chromosomes are called autosomes. Each pair of autosomes is designated with a number from 1 to 22. In general, the numbers have been assigned by the sizes of the chromosomes. Chromosome 1 is the largest, chromosome 2 is the next largest, and so forth. In the middle of each chromosome is the so-called centromere. It is not the exact geographical center of the chromosome, but rather lies closer to one end of the chromosome. We'll discuss the function of the centromere in a later module. The ends of each chromosome are known as telomeres. The karyotype is the number and appearance of chromosomes in a single cell. Here you see the karyotype of a normal human cell with the full collection of 46 normal chromosomes, 2 of which are X chromosomes. It's worth noting that not all human cells have 46 chromosomes. Indeed, pretty much all cancer cells have either less than or more than 46 chromosomes. Having an abnormal number of chromosomes is part of what makes them cancer cells. Here's a karyotype from a human cancer cell. As you can see at a glance, this is nowhere near a normal karyotype. Take chromosome 1. Instead of just being a pair of chromosomes 1, there appear to be 6 copies of chromosome 1. And only one of those copies is normal in size. The other 5 copies either have extra DNA in addition, or are missing part of the chromosome, a deletion. Now look at chromosome 4. There's only one copy, with the other copy missing. This is true of many of the chromosomes, including the sex chromosomes. At the bottom of this picture, you see a bunch of chromosomes that could not be identified. These chromosomes are essentially mashups of pieces of what started as normal chromosomes. With so many abnormalities in this karyotype, it's probably not too surprising that the cell from which it came is very abnormal, that is, a cancer cell. Let's go back to normal chromosomes. As stated earlier, the centromere is not at the exact center of a chromosome, but is closer to one of the chromosome ends, the telomeres. Each part of the chromosome spanning from the centromere to one of the telomeres is called a chromosome arm. By convention, the shorter chromosome arm is designated P, while the longer chromosome arm is designated Q. So when you hear somebody refer to chromosome 1P, it's a reference to the shorter arm of chromosome 1. Each chromosome arm is divided into smaller regions, each of which is designated by a certain number. The numbers used differ from chromosome to chromosome, but there is a common convention, which is that the larger the number, the closer the region is to the telomere. As one example, the region of chromosome 1 called 1p35 is closer to the telomere than the region called 1p13. Flipping it around, 1p13 is closer to the centromere of chromosome 1 compared to 1p35. chromosome regions can be further subdivided, with decimal points used. For example, chromosome 1p13.3 is a smaller region within chromosome 1p13. In a human cell, the 23 chromosome pairs are located in the nucleus, which is enclosed in a membrane that separates it from the rest of the cell. It turns out that the 23 chromosome pairs are not quite the entirety of the genome. There is also a small amount of DNA, relatively speaking, in the cell's mitochondria outside of the nucleus. Let's now switch gears and talk about the mutations that are responsible for diseases and other phenotypes. Mutation is a word that gets thrown around a lot and is used by different people to mean different things. But in general, it signifies a change in genomic DNA. In principle, mutations can represent both small-scale changes and large-scale changes, anything from a single DNA base pair change to a large chromosomal deletion. But typically, one is discussing small-scale changes. The proper term for a small-scale change in genomic DNA is DNA sequence variant. In the nomenclature generally used by human geneticists today, if a DNA sequence variant is found on less than 1% of chromosomes in a population, it's called a mutation. If a DNA sequence variant is found on more than 1% of chromosomes in a population, it's called a polymorphism, which comes from the Greek words for many forms. There are a variety of types of DNA sequence variants. A single nucleotide polymorphism, or SNP, is a DNA sequence variant in which a single DNA base differs from the usual base at that position in the genome. Variable number tandem repeats, or VNTRs, occur when the number of repeats or copies of a short DNA sequence varies. In a copy number variation, or CNV, the number of repeats or copies of a large DNA sequence, greater than 1,000 base pairs in size, varies. Finally, an indel is a special kind of variant where the number of repeats of a sequence is either 0 or 1. The sequence is either present, an insertion, or it's absent, a deletion. Put the words insertion and deletion together, and you get indel. Here's a graphic representation of the various kinds of DNA sequence variants. We start with the original sequence, which geneticists also called the wild-type sequence, or the reference sequence. Compared to the reference DNA sequence, we have a SNP where an AT base pair is changed to a GC base pair. Here we have a VNTR where the trinucleotide CCT, as shown in the box, is present in 2 copies instead of 1 copy. Finally, here we have an indel where the sequence CAC, as indicated by the thick lines, is deleted when compared to the reference sequence. Let's briefly discuss single nucleotide polymorphisms, or SNPs. SNPs usually, though not always, occur in 2 different versions, each being a different base pair. These versions are also known as alleles. Put another way, most SNPs are biallelic. There are 2 different possible alleles, each representing a different base pair. In a given population, one of the SNP alleles will occur more commonly than the other allele. The first SNP allele is called the major allele, and the other allele is the minor allele. Thus, the minor allele is the less common allele in a population. The minor allele frequency, or MAF, of a SNP is the frequency of the minor allele in a population. A SNP with an MAF of 10% has its less common allele occurring on 10% of chromosomes in a population. Or to put it another way, the major allele is present on 90% of chromosomes in a population. The MAF can vary anywhere from less than 1% to as high as 50%. SNPs are very common in the genome. More than 10 million common SNPs have been identified to date. This means that, on average, a SNP occurs every few hundred base pairs in the genome. In contrast to SNPs, VNTRs can be highly polymorphic, having many different alleles in a population. As one example, a well-characterized VNTR that has been given the designation GATA61A06 has been found to have 6 different alleles. In one allele, there are 219 copies of the repeat sequence. In another allele, there are 223 copies of the repeat sequence, and so forth. For VNTRs, the repeat sequence can be quite short, as little as 2 to 6 base pairs. When that's the case, the VNTR is also known as a microsatellite, as single sequence repeats, or as short tandem repeats. As you may be aware, trinucleotide VNTRs, in which the repeat sequence is 3 base pairs in length, have been implicated in a number of diseases, including Huntington's disease and Fragile X syndrome. As we'll see in later modules, VNTRs are particularly useful for family-based linkage studies, whereas SNPs are particularly useful for population-based association studies, most notably the genome-wide association study. Let's close by considering how DNA sequence variants, or mutations, come to exist in the first place. There are 3 major sources of mutations. First, they can occur spontaneously, via the alteration of DNA bases or mistakes in DNA replication. Second, mutations can be induced by chemicals or radiation, which can directly cause the alteration of DNA bases, or even outright breaks in DNA strands. Finally, it turns out that there are so-called transposable elements in the genome, thought to be derived from ancient viruses, that at one point had the ability to jump around the genome, thus serving as another possible cause of DNA alteration. Here's an example of a spontaneously occurring mutation. We start with an adenine-thymine, or AT, base pair. A spontaneous chemical reaction causes the conversion of the adenine base to a non-standard DNA base called hypoxanthine, or inosine, or I for short. At this point, 2 things can happen. The cell has repair machinery designed to find non-standard DNA bases and restore them to the original form. That could occur here. Alternatively, the inosine may go unrepaired. Inosine cannot form a base pair with thymine. Instead, it forms a base pair with cytosine. So it's possible that the cell's repair machinery will try to fix the problem by instead replacing the thymine base with cytosine, thus leaving the inosine in place. Thus, an AT base pair has become an IC base pair. Now that we have an IC base pair, let's see what happens when the cell divides. In DNA replication, the two strands of the DNA molecule will separate, and new strands will be built on the old strands. In one of the new strands, a cytosine will be used to base pair with the existing inosine base. In the other new strand, a guanine will be used to base pair with the cytosine. The ultimate consequence is that an AT base pair has now fully transformed into a GC base pair. And that GC base pair will be transmitted to future generations of cells. Here's a different example in which exposure to a chemical creates a mutation. The chemical here is N-ethyl N-nitrosourea, or ENU for short. We start with a guanine cytosine, or GC, base pair. ENU is an alkylating agent, which means that it can add a carbon side chain to molecules. In this case, ENU reacts with the guanine base. This results in the guanine converting into O6-ethylguanine, or EG for short. EG cannot base pair with cytosine, so if the EG is not repaired, there's the possibility of the cytosine being replaced with thymine, with which EG does base pair. If that should happen, a GC base pair will have become an EGT base pair. Now that we have an EGT base pair, let's see what happens when the cell divides. In DNA replication, the two DNA strands will separate. In one of the new strands, a thymine will be used to base pair with the EG base. In the other new strand, an adenine will be used to base pair with the thymine. Thus, in the end, the original GC base pair has become an AT base pair. So those are some examples of how mutations can arise in a cell's DNA. If that cell happens to be an egg cell or a sperm cell, the mutation can potentially be passed on to a son or daughter, who in turn can pass it on to the next generation, and so forth. The mutation can thus take hold in a human population. In contrast, if the original mutation occurs in a cell type other than an egg or sperm cell, then it will remain within the one person and will not affect any of that person's children. That's not to say that the mutation won't have adverse consequences. For example, it might cause that person to develop cancer, but only that person will be affected.